Welcome to the December webinar on the Glucocorticoid Toxicity Index. I'm Dr. John Stone, Professor of Medicine at Harvard Medical School and the Edward A. Fox Chair in Medicine at the Massachusetts General Hospital. It was my privilege to lead an international group of investigators in the development of the GTI with work that began way back in 2015. This has been a long time in development. So thank you for joining us today. So I want to begin with this quote from a paper which is now under review from our group. In short, glucocorticoid toxicity has hung heavily over the treatment landscape and been an accepted part of the patient experience for those with inflammatory disease for too long. It is time to acknowledge it, to measure it, and to change it. So these are some of our partners in academia and in industry who are helping us, working with us, licensing the GTI on a variety of different inflammatory disorders that I'll show you later. So here's the agenda. I will say a few words first about the GTI as an outcome measure or marketing tool and give a brief regulatory update. There's exciting news in that area. I will talk about data from a phase 3 clinical trial, and before that, we'll give a little bit of a background to the GTI to help you understand the data. Then we will show you a recorded demo illustrating use of the GTI digital platform and entry of data. And then my good friend, Dr. Paul Brunetta from Sana Biotechnology has joined us, and he will talk about use of the target product profile and how the GTI might fit into that. So the GTI, first, is a weighted, validated clinical outcome assessment. It measures change in steroid toxicity. It's not simply a laundry list of all the 75 or so known adverse effects of glucocorticoids. It measures change between 2 points in time. For example, between baseline and measurement of the primary endpoint, or between baseline and some interval period. So the GTI as outcome measure. GTI scores can demonstrate that new drugs are safer and more effective than standard of care. The GTI as marketing tool. GTI scores have demonstrated that new drugs are safer and more effective than standard of care, and data that I will show you related to ANCA-associated vasculitis and also in asthma. So here's a brief regulatory update. Really exciting news in this space. You may have been following this story. Earlier this year, in February of 2021, the New England Journal of Medicine published the Avacopan paper, phase 3 clinical trial of the Aggregate study. This led to the approval worldwide of Avacopan, a novel complement inhibitor for the treatment of ANCA-associated vasculitis. It was approved first in Japan as Tavneos, eventually by the FDA here in the United States, and then most recently in Europe by the EMA. And we're still awaiting wording of the label in Europe, but all this is terrific news. And as I'll show you, the GTI really played an essential role in helping ChemoCentryx establish the efficacy and safety of its drug. So this picture of a patient of mine says all that most of us need to know about steroid toxicity. He's a 19-year-old. He's got a classic moon face. You can see the acne, hirsutism, and he had been on steroids for 2 years. When I met him, he was being treated for ANCA-associated vasculitis. This next picture shows exactly what we want to accomplish. After we had found an effective steroid-sparing therapy for him, you could pass this young man on the street on the right and you would not recognize him from the before picture. This is exactly what we aim for with good steroid-reducing drugs to diminish steroid toxicity. Of course, there are many other phases of steroid toxicity that one can't easily see on the skin, but osteonecrosis and glaucoma and hypertension and diabetes, insufficiency fracture, growth retardation in kids, infection, insomnia, atrial fibrillation, poor wound healing, anxiety. All of these things are well-known complications of glucocorticoids. And another way of framing this is look at this before picture of my patient with ANCA-associated vasculitis, and then this patient here who has lupus, but she might have myasthenia gravis, she might have an inflammatory myopathy, she might have pemphigus vulgaris. These two pictures side by side, which make the two patients look like a brother and sister, underscore the point that no matter what disease we are treating, at some point, we're really managing the same condition in treating inflammatory disease, and that is steroid toxicity. So the GTI has been used in multiple inflammatory disorders now, well-known rheumatologic conditions, ANCA-associated vasculitis, giant cell arteritis, and polymyalgia rheumatica, so the vasculitides, lupus, both systemic lupus erythematosus and lupus nephritis, asthma. We have an excellent, large, real-world longitudinal experience with asthma, pemphigus vulgaris, and inflammatory myopathies and myasthenia gravis, IgG4-related disease, congenital adrenal hyperplasia, ulcerative colitis, sarcoidosis, and in kids, Kawasaki's disease. There's a large European-based trial now of Kawasaki's disease, which uses the pediatric version of the GTI. So our asthma experience was published earlier this year in a couple of different papers, and one of these was accompanied by an editorial from key opinion leaders from New Zealand. The title was "The Vexed Problem of Corticosteroid Toxicity in Asthma, Time for Standardized Assessment." And they made the quote, "The GTI will help prove the clinical significance of novel therapeutic regimens." So the GTI was created by a network of collaborators, experts from multiple specialties: internal medicine, neurology, dermatology, psychiatry, and pediatric subspecialties as well. This network involved experts from all over the world, from North America, from Europe, Australia, and New Zealand. And as I indicated at the top, this work began in 2015. So here are the health domains, and this is getting into the background information that is really required to help you understand the data that I'm going to show you. Here are the health domains addressed by the GTI. So the first four are body mass index, blood pressure, glucose metabolism, and lipid metabolism. And recognize that all of these are easy to measure. They're already measured in the course of a routine clinical trial. These are data that you're already collecting. Bone mineral density can also be collected, although some licensees choose to drop this, but bone mineral density is an excellent measure of steroid toxicity. And then infection, steroid myopathy, skin toxicity, and neuropsychiatric. The pediatric GTI differs only slightly from the adult version in that it has an additional domain, growth, to capture the unwanted impact of glucocorticoids on growth in pediatric patients. So why were these domains chosen? Well, really three simple reasons. First, they are common. These are not rare clinical problems caused by steroids. These are very common adverse effects associated with these medications. Second, they are important. They are not trivial adverse effects. They have a big impact on patients, and providers care about them also. And finally, they're dynamic. In a clinical trial, you don't want to be measuring something that's not going to change. So these domains were chosen because they will change. If the patient is fortunate enough to take fewer milligrams of prednisone or prednisolone or another glucocorticoid over the course of a trial, then steroid toxicity in these domains will diminish. If they have to take more steroid because they're not on an effective regimen, then steroid toxicity will increase. So these domains are dynamic. The GTI provides two different scores from the same set of data. One is called the Cumulative Worsening score, and the second is called the Aggregate Improvement score. These two scores really are opposite sides of the coin, and they capture the full nuances of steroid toxicity. The Aggregate Improvement score, for example, allows us to understand whether there's actually been improvement in steroid toxicity over the course of a clinical trial. So the GTI can measure both worsening and improvement. So a little bit more about the cumulative worsening score, or CWS. It is a running total of steroid toxicity, everything related to a glucocorticoid adverse event that has occurred since the baseline visit. So it's a running total. It captures both permanent and transient adverse effects. If the patient has insomnia for the first 5 months of a trial because of their glucocorticoid, then that needs to be captured as an adverse event, even if the insomnia resolves with longer follow-up. The aggregate improvement score capitalizes on the fact that some steroid toxicities resolve with time. And in particular, most patients, or many patients who are in clinical trials, are not rookies when it comes to steroid toxicity. They've been on steroids before. They will bring some degree of steroid toxicity to their baseline visit. The aggregate improvement score, which permits toxicities that resolve to be removed, allows us to demonstrate that there's actually been improvement in the baseline. So let's turn now to the Advocate data, Avacopan for the treatment of ANCA-associated vasculitis. And I'll show you how the GTI was used in this context. So these two curves show the mean daily prednisone doses for the two groups. Standard of care is in the gray curve. The Avacopan group is illustrated by the maroon curve. So there were different quantities of steroids in these two trial groups. Now, this was the first part of the primary endpoint for this trial. The primary efficacy endpoint at 26 weeks was non-inferiority, comparing the two groups for remission induction. And the Avacopan group was indeed non-inferior to standard of care. What really differentiated these therapies was the lower glucocorticoid toxicity permitted by Avacopan, and that's where the GTI came in. So if one looks at the cumulative worsening score for the GTI, you see how consistently the 8 domains of the GTI reinforce the value of Avacopan. So you see the difference at both 13 and 26 weeks in infections, and your neuropsychiatric toxicity, and skin toxicity, and steroid myopathy, and lipid metabolism, and glucose metabolism at 13 weeks, and body mass index at both 13 and 26 weeks. So 7 of the 8 domains favored Avacopan. The only domain that did not, was a tie, was the blood pressure domain shown here. It was not surprising that blood pressure didn't separate these two treatment groups, because 81% of the patients in the trial had glomerulonephritis. So here are the composite glucocorticoid toxicity scores. This is the cumulative worsening score at both 13 weeks and 26 weeks. And you can see all of these different domains are summed. Lower scores with the GTI are better than high scores. And at both 13 weeks and at 26 weeks, the Avacopan group was statistically superior to standard of care. So these differences were highly statistically significant, but is this clinically significant? So the minimum clinically important difference is 10 points. And the approach to developing the MCID for the GTI was described in this paper published earlier this year in 2021. And so let's take that MCID and actually compare it to the actual GTI scores. So here are the between group differences for the cumulative worsening score. At 13 weeks, the Avacopan group had a score minus 11 points lower compared to standard of care. And at 26 weeks, the difference was 17 points lower in the Avacopan group, both highly statistically significant and clinically important as judged by the MCID. And what about the aggregate improvement score? Again, lower scores are better with the GTI. And at both 13 weeks and at 26 weeks, you can see here that the aggregate improvement score was better, highly statistically better in the Avacopan group compared to standard of care. These were also clinically significant. At 13 weeks, the Avacopan group had an aggregate improvement score 13 points lower than that of the standard care group. And at 26 weeks, the Avacopan group was 12 points lower. So the GTI permitted the advocate investigators to demonstrate that at both 13 weeks and 26 weeks, Avacopan reduced glucocorticoid toxicity compared to standard of care. And this was not only statistically significant, it was clinically important as well. So the investigators reported the GTI as the most important secondary endpoint of efficacy in the trial. And this is table 2 from the New England Journal of Medicine, showing those numbers, those between group differences on the right in the red rectangle. So let's take a look at the GTI in action. We've entered the patient's baseline data. We're going to show you the change in steroid toxicity between baseline and the V2 visit. We've already entered the baseline data. So let's enter the data for the first follow-up visit. Say, hypothetically, this is 13 weeks. This is the main screen, the home page, if you will, of the GTI app. And it is arranged according to a logical flow in a clinical trial visit for a patient. So the first thing that happens in a clinical trial visit is the coordinator will update the patient's medications. The patient's baseline medications were already entered. The patient was on insulin, you can see over on the lower left, at baseline. Now, at the first follow-up visit, the patient is off insulin. His glucose metabolism has improved. In addition, his lisinopril, 20 milligrams at baseline, is now 10. So he has had improvements in both of his medications to treat glucose metabolism and to treat blood pressure. Now we confirm that the intention was to decrease both of those. The vital signs from entry are shown on the left. The patient was 1.64 meters tall at entry and weighed 96 kilograms. The BMI calculated automatically. His weight has improved substantially. He's now down to 89.1 kilograms. His blood pressure has also improved, going from 144 at baseline to 132 over 85. There's been a significant improvement. The hemoglobin A1C is now down to 5.5%, despite the fact that the patient has been able to stop his insulin. His LDL hasn't changed very much. It's 3.1 now in follow-up. So the LDL is now complete. Bone mineral density is a very sensitive indicator of prednisone. So the bone mineral density is 5.1 at the femoral neck and 5.0 at the lumbosacral spine. Now the clinical research coordinator might put the patient into a room. The research coordinator has finished with what he or she needs to do with the patient, checking medicines, vital signs, weighing the patient, etc. The patient goes into an examining room where the clinical investigator examines the patient for myopathy, for skin changes, for neuropsychiatric toxicity, and for infections. So the patient did have myopathy at baseline, but now it has resolved. So up in the upper left, you see that he had a minor steroid myopathy at baseline. The app cues the investigator into what these definitions are. All of the use materials are contained within the app. Now we move to skin effects. So the patient had lots of skin effects at baseline. In the upper left, you see that there was moderate hirsutism, there was a minor acneiform rash, and there were striae that were moderate. There are skin effects now at follow-up. The hirsutism has improved. It is now only minor. And the definitions, again, of each of these grades is included in the app to facilitate rigor. And you can blow up some of these images. Acne has actually resolved. The striae remain moderate. Then we move to the neuropsychiatric domain, and the patient had these manifestations of steroid toxicity at baseline. In the neuropsychiatric domain, there was moderate insomnia and moderate depression. Both of these are now minor at follow-up. Again, the definitions of these neuropsychiatric toxicities are included. And then we go to infections. The patient had oral candidiasis at baseline, 1 episode of oral candidiasis, again, between baseline and week 13. So that completes the changeable elements of the GTI. And now we round out the list of other toxicities by looking at the specific list. And we'll just say that there have been no additional toxicities, but it shows that the patient had a cataract at baseline, and that was carried forward now to this follow-up visit because items in the specific list represent damage, they don't change. Okay, so now we're complete and we can get the first GTI score. Over on the left-hand side, you see the individual domain scores. And then in the upper right, you see the cumulative worsening score and the aggregate improvement score. And what this demonstrates is that there has been a little bit of worsening. The patient did have an episode of oral candidiasis between baseline and week 13, but all in all, there was substantial improvement in steroid toxicity. AIS had actually improved by minus 209 points. And now we will switch over to the target product profile. And it is a great pleasure to introduce Dr. Brunetta, who is a longtime friend and colleague and was actually instrumental in the development of the GTI. He is now at Sana Biotechnology, where he's the head of clinical and translational science. He's going to talk with us about the TPP.
John, it's a pleasure to contribute to this effort, and it's really exciting to see the way the GTI has been changing over time in this presentation as well. I've been in biotech for 20 years and thinking through drug development, thinking through toxicity, and aware that I think the GTI represents a form of precision safety monitoring for some of the most important adverse events that are part of our assessment of those therapies where corticosteroids have been part of a standard of care, where the adverse events have on some level been accepted because there weren't good alternatives. And now what you see is that there are some very, very important drugs that are important alternatives. And to measure the reduction in corticosteroid toxicity is truly a measure of the benefit of that drug. So if we go to the next slide, I thought what I'd do would be to walk people through what is a TPP. It's hard for me to imagine working through drug development without having a target product profile. It really defines success, what that looks like in the clinic. It anchors very good decision-making across multiple different areas of a biotech or a pharma company through research, development, commercial aspects of how a therapy might be defined. It's typically really reviewing the efficacy measures, the safety outcomes, the dosing and administration, and on some level, which I'll show you, is basically the one page that all of these different functions come around to understand what is the benefit of the therapy that we're working to put into the clinic for patients, and that's tested through a clinical development plan. Now, obviously, new information could come in that might modify a target product profile, new safety information, phase 2 results, competitor information, a change in the entire field. But what was occurring to me is that if there were ways that the glucocorticoid toxicity index were considered to be included in a target product profile over the course of a clinical development program, it could really help a company capture the potential benefit of a therapy in reducing steroid-related toxicity as steroid use is also reduced. Again, the key elements are efficacy, safety, dosage, and administration. The way to create a TPP basically is to start off with a group of individuals that are focused on the life cycle of a therapy and the development of a therapy, and you start off with some basic features. It's draft. It changes over time. It involves all parts of an organization. So it's something that is clear and simple enough to understand if you're a scientist or if you're a non-scientist. Perhaps you work in regulatory, you may not have a scientific background. Perhaps you're in research, you may not have a commercial background. So it represents a clear and simple way of understanding what are we trying to do with this particular therapy. And over the course of a clinical development program, which can last for many years, that target product profile is present it to patients, to physicians, to payers, to healthcare systems to get feedback. Would you see this as a valuable new entrant in the treatment of these patients? Again, it can evolve over time with new information, and really can also help to define, go, no go criteria. If you really feel you're hitting the mark in your research as it's unfolding, or if you have a major issue that suggests you're not going to be in the space that you think would be of the best benefit. For instance, if your efficacy isn't above 50% as measured by a particular efficacy outcome, we may not have a competitive product because there are already other products in that space. So it can help to set up some bars or some decision-making because you've gotten feedback on the clarity of what you're trying to create over time. There is FDA draft guidance related to the target product profile. So certainly the regulators understand that this can be very important to use. And there was a publication in 2017 suggesting that it may not be used as often as probably it should be, because it can give you specificity and clarity of what your target is over time. And, perhaps not all companies are using it the way that they might. The next slide shows you just a basic example of what a target product profile would look like. Typically, you have an indication statement that says drug X is indicated for the treatment of a particular disease. Efficacy measures, as that include the primary endpoint or secondary endpoints. Safety measures that might have some criteria related to the safety profile that would be acceptable in that particular disease. And then dosing and administration. And we often then break it down into target. What is the target that you're specifically focused on achieving? There might be optimistic or optimal levels that even exceeds those targets for what you'd hope for efficacy and safety. And there may also be some minimal criteria where if you don't achieve these minimal criteria, then the therapy that's being developed may not be really a benefit or provide the benefit that was anticipated when it was first started. The next slide shows you what might be considered in this case with the glucocorticoid toxicity index. So as this instrument is being applied, and Dr. Stone had shown you how it might be applied during the conduct of a clinical research study, in the safety elements, if you were to say that a particular drug significantly reduces glucocorticoid toxicity above the minimally clinically important difference, that could be considered either in the optimistic or in the target profile for a particular therapy. It doesn't necessarily have to be in the minimal. So for instance, if a therapy is beneficial from an efficacy perspective, it has a very good safety profile, the benefit and risk are in favor of the use of that therapy, but it didn't achieve the reduction in glucocorticoid toxicity that might have been target, it doesn't mean that it isn't still a significant therapy. So this can be managed and understood during the course of a clinical development program, phase 1, phase 2, and phase 3. Especially in autoimmune or inflammatory diseases, these are often not extremely small studies. They're often randomized control trials, which is where the GTI has largely been intended to be used. So I think this could be a way in which it would be managed and modeled over time and potentially incorporated more into clinical development programs for novel therapies where part of the true benefit of that therapy is the potential to reduce corticosteroid toxicity. And that's the main point that I'm trying to make. This last slide is really just intended to show you the many different stages of drug development. Obviously, early discovery, showing that there's proof of biology, proof of concept for a therapeutic pathway. Early stage research where you may be and would be considering early in vivo or animal studies to assess toxicity. Then thinking through phase 1 studies, phase 2 studies into marketing. And the target product profile is typically generated in early to late stage research during that period where you're showing efficacy either in the lab or in animal studies that is going to start to target that drug to a specific disease and perhaps a specific patient subset in that disease. Around that time, you might generate a target product profile and then have it be used throughout the course of early and late stage clinical development. And then those results can really be moved forward into the way you would generate a draft product label. So if you do achieve the milestones, if you do have target efficacy, safety, dosing, administration, that information can then lead into the way you would think about bringing forward a label for a therapy that can get approved by the FDA or by agencies around the world. And obviously, having an excellent safety profile, having the right data, having a validated instrument that has a track record, all those are really important aspects of showing that there is a benefit as it relates to a reduction in steroid toxicity, but it's also a reproducible benefit that is really credible. And I think that's what John was suggesting and showing with the Avacopan data. And I think there are multiple opportunities to consider this across many different diseases and many different new biologic therapies that are coming forward. So I'll end there, John, and any other comments or suggestions?
Thank you for introducing me to the topic of TPP. Before we sort of move on to conclusion and take questions through the chat, I just had a question for you. Which part of a company typically initiates TPP development? Is it usually clinical, medical affairs, or some other part of the company that takes the lead on this?
You know, everything we do is through cross-functional teams. So there are cross-functional teams that include research, discovery, but also can include clinical, commercial, even in the earliest parts of early stage research, right? Because if you're going to be targeting a drug, you're going to want to understand, well, what is that patient population and what are the current therapies for that patient population? So it's multiple different functions that are part of cross-functional teams all the way through early stage research into the post-marketing setting and medical affairs. Now, early research is far more scientific. They may be viewing people in later stage research more as consultants. But as you get into phase 1, phase 2, phase 3 studies, again, you have much more of the clinical and commercial input into how would this therapy actually be used and what benefit does it represent?
I've always been impressed by how good companies manage to keep everybody on message, like they're singing from the same hymnal. And this may be one of the ways that they do that.
It is, John. It's really the one pager. You know, it's the way that we all understand where are we going and what benefit do we want to provide for patients as it relates to efficacy, safety, and dosing. And if we have real clarity of the benefit in safety, that's critical. It shouldn't just be something we observe. It's something that I think the instrument is really beginning to clarify what the actual benefit would be. So that's what the TPP is. It's the one pager through years of development that really shows the benefit that we're trying to derive from a new therapy.
Thank you very much, Paul. I just want to sort of sum up here. The GTI simplifies. It uses data that are already being collected in most trials, and investigators are prompted to make a few additional observations that are important to assessing steroid toxicity. The GTI standardizes scoring across investigators and sites. It focuses on common, important, dynamic domains of steroid toxicity, and it measures change in steroid toxicity, allowing for the possibility that steroid toxicity may improve as well as worsen. So thank you very much for your attention. Paul, thank you very much for your collaboration. Thank you very much.